Hello, welcome back to my YouTube channel. In this video, I'm going to talk about RNA sequencing data analysis. I really wanted to make a series of tutorial where I will explain all the steps of RNA sequencing. This series of tutorial is good for everyone, even if you have no background in programming, you have not studied bioinformatics or computer science, still you can follow my tutorials. I will take you from the very basic and I will go a little bit to the advanced level. So first of all, I would like to talk about RNA sequencing. What RNA sequencing is? Why do we need RNA sequencing? What are the steps of RNA sequencing? So the first we can talk the RNA sequencing. Basically, RNA sequencing is a type of sequencing where we sequence RNA. And then we can talk why do we need RNA sequencing. Most of the time, you need RNA sequencing because you want to understand the expression level of the genes. For example, when you are trying to sequence RNA, it means you want to study what is happening on the transcriptomic level. And on transcriptomic level, we have the gene. So most importantly, we use RNA sequencing for expression analysis. Like we want to see if there are two types of cell, for example, healthy and disease cell, which gene is expressed in normal cell and which gene is overexpressed or less expressed in disease cell. So this is what we normally do, but of course you can use RNA sequencing for different purposes like annotation. Even you can use RNA sequencing data for drug discovery. Now I want to talk about the RNA sequencing. When we are going to perform RNA sequencing data analysis, what are the steps that we should know? The first thing is we need to extract the RNA from the respective sample. It can be human cell lines, mice cell lines, can be plant tissue or anything. Like you can use any sample and you can extract the RNA. Once you have extracted the RNA, then you can prepare the libraries. After preparing libraries, you do the sequencing and then data analysis. So these are the 4 steps here that you have to follow if you want to do RNA sequencing data analysis and you want to study expression of different genes. As you guys know, this tutorial is RNA sequencing data analysis tutorial. So I will talk about the steps involved in the data analysis of RNA sequencing. I will skip the first 3 experimental steps like RNA extraction, library preparation, and sequencing. For data analysis part, we get the data from the sequencing machine. To perform quality control, we use fastq files generated by sequencer, and we do 2 things. The first is we perform the first step of the quality control using fastq package. This package is available on command line tools, and then we trim the adapter. When we perform library preparation, we attach the adapter because we have RNA, it's like a big sequence and we don't want to sequence it like whole. What we do is we make small fragments, and each fragment we add a specific adapter with a sequence. So these adapter has to be removed in the downstream analysis. So the first step is quality control, and in quality control, we do quality control using fastq. And for the second step, we remove the adapter, and we can remove the adapter with cut adapt or flex bar. I will use the cut adapt because I'm gonna use the terminal on Mac or Linux system. I don't have Ubuntu. In Ubuntu, you can use the flex bar. So the next step in the data analysis part is alignment and mapping. In alignment and mapping, there are 3 stages. In the first stage, we generate the index using star package. Star package is also a conta-based package. In star help us to generate the indexes. Indexes is actually a way where we use annotated file and our reference genome to use the indexes. And after that, we map our reference genome with our reads the reads that we got from the sequencing machine. And once we are done with these two steps, we go for the third step, that is count. For count, we use feature count. I will explain all these things whenever we are performing. I will talk about what are the input file, what are the output file, what commands I'm using, and what does each file mean, and what is the information in each file. I will explain each and everything. So the next step is performing differentially gene expression analysis, which we will perform on R. And for R, we have a library DSeq, which will help us to perform this analysis. Here, I would like to show you a diagram which will give you a more comprehensive perspective to the RNA sequencing data analysis steps and the packages that we use in each step. So here you can see, like when we are performing the RNA sequencing, we need three kind of file. The first file is raw sequencing data that we get for the sequencer, then the annotation file and reference genome. So these file we get from the sequencing machine, annotation and reference genome, we can download from different databases. Once we have these files, we can go for the pre-processing, which is also called the quality. In quality, we do trimming and indexing, or we use fastq to generate quality control reports. Once we've done that, we can use Tophet, Star, or any other software to do the alignment. And after that, we can go for the assembly, and then it's like a simplified or a more comprehensive list where you can see the different kind of packages that are developed on different tools which you can use in RNA sequencing. So this is just a brief introduction, but I will guide you for each step, starting from the downloading data and then performing the quality control, then alignment and mapping, and after that, differentially gene expression analysis. So here I want to talk about the system requirement. If you want to do data analysis, you need to have a special system. It's not the normal system that you can use like your Windows system. You need Linux-based system, Linux or Unix-based system. The first recommendation from my side is you can use the MacBook Pro M2 16 GB RAM. This is what I'm using. This is very good. You can handle different kind of data, and this machine is good, but it's also not really good if we have a big data. For example, if we get fastq files from sequencing machine and each file is 1 to 2 GB, then my machine is also not really good. Second recommendation is you use virtual machine. I will make a tutorial how you can sign up for Amazon or Microsoft Azure virtual machine. Like for example, you can get their trial, you can sign up for the trial, and then you can use it to create machine, and you don't have to pay the money, because you will get few features of cloud computing in the trial. So this tutorial can be covered within that virtual machine. And then there is a Ubuntu system. Ubuntu is a kind of Unix like system which you can install in Windows. It's a kind of operating system that you can use, but in that case, if you install Ubuntu in your Windows laptop, then whenever you are turning on, like whenever you reboot your system, you will get two options: get into the window or Ubuntu. Ubuntu you can use for data analysis part, and window you can use for your normal daily life. So these kind of systems can be used for RNA sequencing data analysis, but most of the companies or research centers, they use virtual machines because virtual machines are really easy to handle. Microsoft or Amazon cloud computing, you can pay like 100 or 200 euro, and you can use if you, for example, you have one virtual machine, that virtual machine can be used by multiple people. So thank you very much for watching this video. Next video is system setup and data downloading. If you like the video, subscribe it, like it, and share with your friends. Bye bye. Take care.